I want you to try and imagine something for a second. Imagine the exact moment you finish your work for the day.
Yeah.
You close the laptop, you put your phone away in another room, you sit down on the couch, and you just try to do absolutely nothing.
Right. Total rest.
Exactly. Total, absolute rest. But what actually happens, I mean, if you're like most people, your brain doesn't quietly shut down. Instead, it gets incredibly loud.
Oh, absolutely.
Memories just start bubbling up out of nowhere. You start rehearsing conversations you might have tomorrow, or you replay some incredibly awkward thing you said like 3 years ago. You try to rest, and your mind just takes off running.
Yeah, it is a nearly universal human experience.
Yeah.
And historically, we've really viewed the brain as this machine that we consciously drive.
Like a car.
Like a car, yeah. The assumption was always that when you take your hands off the steering wheel, the car simply idles quietly. But, what neuroimaging and modern molecular biology are revealing is that the real secret to our identity, to our mental health and our resilience, it actually lies in the networks that operate entirely in the background.
The ones running while we're supposedly idling.
Precisely. These are the networks that seize control the millisecond you let go of the wheel.
And that is exactly our mission for this deep dive today. We have an incredible stack of sources spanning across cognitive neuroscience, molecular biology, and behavioral psychology.
It's a huge stack.
It really is. We are going way down the rabbit hole to uncover the hidden architecture of the mind. We're going to explore what your brain is structurally doing when it's supposedly at rest. We'll look at how it builds these rigid habits on autopilot, how it physically processes mistakes like at a cellular level, and finally, how your life experiences literally rewrite your biology to make you resilient.
And, the stakes here are quite high. We are talking about the very biological foundations of who you are as an individual.
Right.
Understanding these background processes, it really shifts how we view mental health disorders. It reframes our understanding of trauma. And most importantly, it gives us a blueprint for how we can actively build biological resilience rather than, just waiting for it to develop.
Okay, let's unpack this.
Yeah.
We have to start at the baseline, right? The foundation of how we process the world when we aren't actively trying to complete a specific external task.
Yes, the resting state.
We are talking about the default mode network, or the DMN. And to really grasp how fundamental this network is, we have to look at the history of how science got the resting brain just completely wrong for, the better part of a century.
Oh, absolutely. The initial misunderstanding was profound. If we go back to 1929, there was a scientist named Hans Berger, and he invented the electroencephalogram, the EEG.
Okay, the EEG. That's the one with all the wires they stick to your scalp.
Yeah, exactly. He was the very first person to measure the electrical activity of the intact human brain. And Berger noticed something completely unexpected. The electrical oscillations, the brain waves, they didn't stop or flatten out when a person was just sitting quietly with their eyes closed.
They kept going.
They kept going. So he proposed that the brain is constantly active all the time, but the neurological community of the era essentially dismissed him.
Wait, really? They just ignored the data?
Pretty much. The prevailing assumption back then was what we might call the light bulb theory. It's the idea that the brain only turns on or becomes metabolically active when you are doing a specific, focused mental task.
Okay, which brings us to the Sokoloff study from the 1950s that I was reading in our notes. I was fully expecting his data to support that light bulb theory.
Most people do.
Right, because Louis Sokoloff was measuring how much metabolic energy the brain consumes. He measured people sitting quietly at rest, and then he made them do these really effortful, complex math problems.
Very difficult math, yeah.
And I assumed the math problems would light the brain's energy consumption up like a Christmas tree. But his metabolic data showed, almost no change. I actually thought I was misreading the chart.
It's shocking the first time you see it.
How is it physically possible that doing complex calculus burns almost the exact same amount of calories as just staring blankly at a wall?
Yeah, it is entirely counterintuitive. The metabolic rate basically stayed within a tiny margin of error. But if we fast forward to the 1990s, with the invention of PET scans, positron emission tomography, researchers finally had the tools to track blood flow and see which specific areas of the brain were working hard.
They saw a different pattern.
A very consistent, bizarre pattern. When you give someone a highly demanding cognitive task to do like a complex language puzzle or a visual search, certain specific areas of the brain actually become less active than when they were just resting passively.
So the brain was actively powering down certain regions to focus on the external world.
Correct. And then Marcus Rachel and his lab at Washington University, they put all these pieces together around 2001, and he coined the term default mode.
No. Yeah.
He identified this highly specific, interconnected network of brain regions that are intensely active when we are at wakeful rest and that synchronously deactivate when we focus outward.
And the statistic from Rachel's lab in these sources is just staggering. When you sit down and do a demanding mental task, your brain's total energy consumption goes up by less than 5% compared to its baseline resting state.
Less than 5%.
Less than 5%. I mean, if the brain accounts for roughly 20% of the body's entire energy consumption, and the vast majority of that is dedicated to just sitting there, what on earth is it doing with all that fuel?
Well, that 20% figure is massive when you consider the brain is only about 2% of your body weight.
Right.
Now, a significant portion of that baseline energy is used for simple physiological maintenance. Maintaining resting membrane potentials across billions of neurons, recycling neurotransmitters, doing synaptic pruning.
The house cleaning stuff.
Exactly. But a huge fraction of that energy is actively powering the default mode network.
So if I can try to refine my car analogy here, it's not just that the brain is a car engine idling quietly in the driveway. It's more like the brain is this massive data center.
Oh, I like that.
Yeah, even when you aren't actively running a specific search query on your monitor, the servers in the background are still drawing massive amounts of power to, I don't know, index files, run background simulations, and update the operating system.
That's a much more accurate way to look at it.
So where are these servers actually located in the brain?
Anatomically, the default mode network is composed of a few key hubs connected by these thick tracts of white matter. You have the medial prefrontal cortex, which sits right behind the center of your forehead.
Okay, front of the brain.
Right. Then you have the posterior cingulate cortex and the precuneus, which are tucked much deeper in the brain toward the back. And you have the angular gyrus on the sides in the parietal lobe.
Okay, so it's really spread out.
It is. Originally, scientists called this the task-negative network because these areas powered down during external tasks. But neuroscientists have largely abandoned that term because it heavily implies the network is doing nothing.
And it's definitely not doing nothing.
Not at all. It is actually dedicated to highly complex internal tasks.
The inner storyteller.
Exactly.
If this network is burning all this energy while we are supposedly at rest, it's constructing a narrative. It's doing the heavy lifting of autobiographical processing.
Yes. The DMN is responsible for a suite of very specific cognitive functions. It activates heavily when you are remembering the past, retrieving episodic memories and placing yourself back in a specific time and place.
Okay.
It activates when you are engaging in prospection, which is imagining the future, running simulations of what might happen tomorrow so you can prepare for it. It activates when you reflect on your own emotional state.
So it's very me-focused.
Yeah, mostly, yes. But crucially, it is also highly active when you are thinking about the minds of others. Psychologists call this theory of mind the ability to infer what someone else is thinking, feeling, or planning.
Which brings up some of the most fascinating neuroimaging studies in our source stack. Researchers looked at what happens to the DMN when we consume stories.
Yes, the narrative syncing studies.
Right, when they put people in an fMRI scanner and have them listen to an audio story or watch a movie, their default mode networks sync up. If you and I are listening to the same gripping narrative, our DMNs are firing in the exact same correlated patterns, second by second.
And the methodological specificity of that research is really what makes it so robust. The researchers didn't just play audio, they manipulated the narrative to see what the brain was actually reacting to.
Right.
For instance, if they take the audio story and scramble it, playing the sentences out of order so it sounds like coherent English but lacks any real narrative arc, the DMNs of the listeners do not sync. And if they play the story in a language the listener doesn't understand, the auditory cortex fires, obviously, but the DMNs don't sync.
So it's not just reacting to the acoustic properties of a human voice.
Not at all. And to prove that, they presented the exact same conceptual story to different people in different languages.
This is the part that blew my mind.
They had Russian speakers listen to a Russian translation, and English speakers listen to the English version. And as long as the listeners understood their respective languages, their DMNs synced up perfectly across the two groups.
Wait, I have to push back on this for a second.
Go ahead.
If the DMN is the me network, the exact neural circuitry that burns fuel to build my internal autobiography, my specific memories, my future plans, why does it perfectly sync up with someone else's brain when we listen to a completely fictional external story?
That's a great question.
It seems like a huge contradiction. Why would my self-network care about a fictional character?
It does seem like a paradox, but it actually reveals the fundamental nature of human cognition. The DMN isn't just a static filing cabinet for facts about you. It is a dynamic narrative engine.
A narrative engine.
Yes, we fundamentally understand our own lives as a story. We view ourselves as the protagonist with characters, motivations, past traumas, and future goals.
Oh, I see.
So when we hear a compelling external narrative, we don't have some separate, dedicated fiction processing network. We use that exact same autobiographical neural machinery to comprehend the story.
That makes so much sense.
We transcend the auditory processing of the words and map the conceptual meaning using our own internal framework.
We literally use our self-network to understand the other.
Precisely. We are mapping their simulated mental states onto our own circuitry.
That reframes so much about how we connect with art, and links perfectly to that 2019 study by Edward Vessel, G. Gabriel Starr, and their colleagues in our notes.
Ah, yes, the neuroscience paper.
Right. They found the DMN is deeply tied to how we perceive beauty and aesthetic pleasure. When participants were in the scanner looking at pieces of art, the visual cortex obviously activated.
Naturally.
But when they saw a piece of art that they reported as deeply, personally moving something they found truly beautiful, the DMN suddenly lit up, and coupled with the visual network, it's like we are pulling that external art into our personal identity.
Exactly. Beauty in this neurological context isn't just an objective calculation of symmetry or color balance happening in the visual cortex.
Right, it's not just math.
It is a profound resonance with our internal narrative. The art triggers a sense of self-relevance, but, we must consider the darker implications of this as well.
The dark side of the DMN.
Yes. If this narrative engine is so powerful and so metabolically demanding, what happens when the story it tells goes wrong?
Right, because when we sit alone in a room, the stories our brains spin aren't always beautiful or helpful. Sometimes they're awful, which brings us to the clinical vulnerabilities of the DMN. If this network uses so much energy, it must be susceptible to wear and tear over time.
It is highly vulnerable. Let's examine Alzheimer's disease, for example. One of the primary biological hallmarks of Alzheimer's is the buildup of amyloid beta plaques in the brain tissue.
Right, the plaques that disrupt memory.
Correct. For years, researchers mapped where these plaques appear first. Randy Buckner and his colleagues discovered that amyloid beta deposition begins specifically in the hubs of the default mode network.
So the posterior cingulate cortex and precuneus.
Exactly there. And this buildup happens long before individuals show any outward cognitive symptoms or episodic memory loss.
But why there? Why does the plaque target the DMN hubs first, before the rest of the brain?
The leading hypothesis is metabolic exhaustion.
Like it just burns out.
Basically, yes. Because the DMN operates at a continuously extraordinarily high metabolic rate throughout our entire lives, these specific regions experience massive amounts of synaptic activity. During normal synaptic transmission, a protein called amyloid precursor protein, or APP, is routinely cleaved. With the DMN constantly firing, the sheer volume of this activity causes the cleaved peptides to accumulate faster than the brain's waste clearance systems can remove them.
Wow, so it's a waste management problem.
Over decades, yes. They clump together into plaques that physically disrupt the network, which eventually leads to the severe episodic memory loss characteristic of the disease.
The network literally burns itself out from decades of overuse. That is terrifying.
It is sobering.
What about neurodevelopmental conditions? The sources also mention autism spectrum disorder.
In autistic individuals, neuroimaging frequently reveals structural and functional differences in connectivity between the hubs of the DMN.
How so?
Specifically, there is often reduced functional connectivity between the medial prefrontal cortex, that frontal hub involved in thinking about the self and others, and the posterior cingulate cortex in the back.
Okay, so the front and back aren't talking as efficiently.
Right. And because the DMN is the primary engine for theory of mind and social evaluations, these distinct connectivity patterns map very closely onto the varying communication styles, social interaction differences, and internal processing mechanisms we observe in autism.
That makes a lot of sense. But the clinical connection that really jumps off the page in these sources is major depressive disorder.
Yes, MDD is deeply tied to this.
We discussed how the DMN kicks into gear when we let our minds wander. Well, in depression, One of the most debilitating symptoms is rumination.
The endless looping.
Exactly, where a person endlessly loops negative thoughts about themselves, analyzing a past failure over and over. And the data indicates that in depression, the DMN exhibits hyperconnectivity.
It exhibits severe hyperconnectivity and a failure to deactivate.
A failure to deactivate, meaning it won't shut off.
Exactly. The narrative engine essentially goes into overdrive and locks onto a negative storyline. It dominates the brain's overall activity, even when the person is actively attempting to focus on an external task.
It just won't let go.
Right. It filters how a person perceives external events, coloring absolutely everything through this hyperactive, self-referential negative loop. And we actually see a similar but distinct dysregulation in ADHD.
Really? How does it manifest in ADHD?
Well, in a neurotypical brain, there is a strong anticorrelation between the DMN and the task-positive networks.
Meaning when one goes up, the other goes down.
Correct. When you focus intensely on a puzzle, the DMN powers down. But in ADHD, this anticorrelation is reduced. The DMN doesn't power down efficiently when the person tries to focus.
Oh, I see.
So the internal daydreaming network and the external focus network are essentially competing for resources at the exact same time.
That sounds exhausting.
It is. This constant intrusion of background processing particularly interferes with the ability to encode long-term memories and sustain attention.
So, to use a computer analogy, again, rumination and depression, is basically the brain's replay function getting stuck in an infinite loop. And you can't force quit the program because the DMN has secured administrator privileges.
That is a highly accurate way to visualize it.
It's dominating the rest of the brain, and the background program is eating up all the available RAM.
Yes. And it explains why treating these disorders is so notoriously difficult. You aren't just trying to talk someone out of a bad mood, you know.
Right, it's not a character flaw.
No, you are attempting to physically alter the resting baseline of the brain's most dominant energy-intensive network.
But the neurobiology also shows us that we can modulate the DMN.
Right. We can,
Because simply telling someone with severe depression to just stop overthinking clearly doesn't work. The biological momentum is way too strong.
We have several biological and behavioral levers. On the pharmacological side, recent clinical trials utilizing psychedelics, specifically psilocybin and LSD, reveal something really extraordinary.
I was hoping we'd get to this.
Functional MRI scans show that under the acute influence of these substances, blood flow to the DMN hubs is significantly reduced, and the network functionally desynchronizes.
Wait, what is the physical mechanism there? How does a chemical like psilocybin actually break apart a brain network?
Psilocybin acts as an agonist primarily at the 5-HT2A serotonin receptors.
Okay, 5-HT2A.
Yes. These receptors are densely clustered on pyramidal cells in the cortex, particularly within the hubs of the DMN. By heavily stimulating these receptors, the psychedelic causes these neurons to fire asynchronously.
So they thought firing it together.
Exactly. It disrupts the rhythmic, coordinated oscillations that usually bind the posterior cingulate cortex to the medial prefrontal cortex. It temporarily shatters the narrative engine.
Wow. And what does that feel like to the person?
Subjectively, patients often report this as a profound dissolution of the ego. Clinically, it provides a crucial window where the rigid loops of rumination are broken, allowing for new neural pathways to form during therapy.
That makes total sense. You are chemically uncoupling the servers.
Precisely.
On a totally different note, the sources mention sleep deprivation weakens DMN connectivity too, though obviously inducing insomnia isn't a sustainable psychiatric treatment.
No, definitely not recommended.
But there's a behavioral lever too. Meditation. The sources detail a landmark study from the University of Massachusetts looking at an 8-week mindfulness-based stress reduction program, or MBSR.
The MBSR study is vital because it proves we can alter this architecture without any pharmacological intervention.
So just with practice.
Right. Participants engaged in about 27 minutes of daily mindfulness meditation for 8 weeks. And the researchers didn't just rely on subjective questionnaires asking if people felt less stressed.
They actually scan them.
They put them in MRI scanners before and after the 8-week period to measure cortical thickness.
And they found actual structural changes in the gray matter just from 27 minutes a day of sitting and focusing on the breath.
Yes. The participants showed increased gray matter density in the hippocampus, which is a region crucial for learning, memory, and emotional regulation.
Okay, so the memory center got thicker.
Simultaneously, they exhibited decreased gray matter density in the amygdala, the almond-shaped cluster deep in the brain that drives the fear and stress response.
So the fear center shrank. That is incredible.
And when we look at functional connectivity in long-term meditation practitioners, We see chronically reduced activation of the DMN. By continuously practicing the cognitive action of noticing a wandering thought and gently bringing attention back to the present moment, they are biologically training their brains to turn down the volume of that background narrative.
Okay, so we've mapped out the wandering narrative mind. We know how it builds our identity, how it can trap us in rumination, and how we can modulate it. But, we don't spend our entire lives just sitting on the couch imagining the future or analyzing the past.
No, we have to interact with the world.
Exactly. We have to make coffee, drive to work, tie our shoes. How do we get through our day without our DMN constantly narrating and overthinking every single physical step we take?
To navigate the physical world efficiently, we rely on a completely different neural architecture, the architecture of habits. When we need to act, we shift from the highly deliberative resting narrative networks to the automated sensoromotor networks. We put our behavior on autopilot to conserve cognitive energy.
And this brings us to a massive shift in how science understands habit formation. Guided by the research from MIT, specifically the work of neuroscientists Kyle Smith and Anne Graybiel.
Yes, their work is foundational.
Because for the longest time, the psychological framework for habits was purely based on behaviorism, right? The
Yep.
stimulus-response model.
Yeah.
Like a bell rings, the dog salivates, you see a red traffic light, your foot hits the brake.
The historical stimulus response, or SR framework, rooted in the work of Pavlov and Skinner, was incredibly influential. It contrasted habits against action-outcome behaviors.
What's the difference between the two?
An action-outcome behavior is highly goal-directed and relies on the prefrontal cortex. You want a piece of chocolate, so you consciously walk to the kitchen and open the cupboard. You are thinking about the value of the outcome.
Right.
A stimulus response habit, however, means you walk into the kitchen, see the visual cue of the cupboard, and open it automatically, even if you know the chocolate isn't there anymore.
Oh, I've definitely done that.
We all have. The behavior has become neurologically divorced from the actual value of the outcome.
But the internal neural recordings from the MIT researchers show that the brain is doing something much more sophisticated than just wiring a trigger to a physical reflex, right?
Huh.
They look deeply into a subcortical brain region called the dorsolateral striatum, or DLS.
The DLS is a canonical habit learning region located deep within the basal ganglia. To uncover its specific function, the researchers used microelectrode arrays implanted directly into the brains of rats.
To measure the electrical spikes.
Exactly. They trained these rats to run a simple T-shaped maze. The rat waits at a starting gate, hears a warning click, the gate opens, and the rat runs down the straightaway.
Standard lab rat stuff.
Partway down, it hears a specific tone, say, a high pitch telling it to turn left for chocolate milk, or a low pitch telling it to turn right for sugar water. Over weeks of repetitive training, the rats become incredibly fast and highly accurate. The deliberation stops. The maze run becomes an ingrained automatic habit.
And while the rats are learning this, the researchers are recording the electrical spikes of individual neurons in the DLS in real time. And they discovered a phenomenon called action chunking.
This was a paradigm-shifting discovery in behavioral neuroscience. You might logically assume that as the habit gets stronger and more automatic, the neurons in the DLS would fire continuously throughout the entire maze run, essentially acting as the motor driving the rat forward.
Right. Like holding down the gas pedal.
But that is not what happens at all. As the behavior transitions from a conscious choice to a habit, the neurons inside the DLS alter their firing pattern drastically. They burst heavily at the very beginning of the maze run when the gate opens. They go almost completely quiet during the actual running and turning, and then they burst again at the very end of the run when the rat reaches the goal.
Here's where the mechanism gets really interesting to me. It's exactly like computer code brackets. Well, when you write a function in programming, you put an opening bracket at the start and a closing bracket at the end. Everything inside those brackets executes as one single continuous command. The DLS is basically putting a start tag and an end tag on a complex physical behavior, chunking it into one unbreakable file.
That is an excellent mechanical analogy. By bracketing the action, the basal ganglia takes a massive load off the prefrontal cortex. The brain doesn't have to make micro decisions at every step of the maze, checking the floor, listening to the tone, calculating the turn.
Just runs the script.
Exactly. And we can physically observe this shift in the rat's behavior. Early in learning, a rat will exhibit what researchers call vicarious trial and error, or VTE.
What does that look like?
They will run to the T-junction, stop entirely, look left, look right, physically deliberate, and then make a choice. But the MIT researchers found a direct quantifiable correlation. The stronger that DLS chunking pattern fires at the very start of the run, the less likely the rat is to pause and deliberate later in the maze.
The autopilot fully engages at the starting line, and it completely overrides any need to think at the intersection.
Exactly.
But the DLS isn't just a rogue agent, right? There has to be a higher-level manager in the cortex that permits this chunking to happen in the first place.
Yes, and that is the infralimbic cortex, or IL. While the DLS is doing the mechanical chunking down in the basal ganglia, the IL, which is part of the prefrontal cortex, acts as the strategy scheduler.
The manager.
The manager. And the researchers proved this relationship using optogenetics, which is a technique that allows precise control over specific neurons using light.
Wait, pause for a second. Optogenetics constantly comes up in modern neuroscience, and the source is mentioned using algae-derived proteins. How do you actually get a light-sensitive protein from algae into the prefrontal cortex of a living rat?
It is a remarkable technique. Scientists take the genetic code for a light-sensitive ion channel found in certain species of green algae, a protein called channel rhodopsin. They insert this genetic code into a harmless viral vector, usually an adeno-associated virus. They inject this virus precisely into the infralimbic cortex of the rat.
Wait, they infect the rat with a virus.
Right, a harmless one, just to deliver the genetic instructions. The virus infects the neurons and inserts the code, causing those specific brain cells to grow these light-sensitive channels on their surface.
That is sci-fi level stuff.
It really is. Then, researchers surgically implant a tiny fiber optic cable into the brain. By shining a specific wavelength of blue light down the cable, they can force the channel rhodopsin to open, instantly depolarizing and activating the neuron. Turn the light off, and the neuron stops firing.
So they literally install a microscopic light switch on the habit-controlling neurons.
Exactly. And the behavioral results were profound. They took rats that were entirely overtrained rats whose DLS had fully chunked the maze habit. While the rat was running, they shined the light to instantly inhibit the IL cortex.
They turned off the manager?
And the rats immediately dropped their newly ingrained routines and reverted back to older, discarded habits or began deliberately exploring the maze again.
Wow.
If the IL cortex is the factory manager, it is deciding which chunk of code the DLS assembly line is allowed to run. It actively suppresses the old strategies and promotes the new habit file.
Okay, so we have the IL cortex saying, run this routine.
Right.
And we have the DLS putting the start and end brackets on the action. But here's the biological mystery that we need to resolve.
Okay.
If the DLS is just blindly executing the chunked action from start to finish without deliberating, how does the brain know if the habit was actually successful? I mean, how does it monitor for errors if it's running a closed loop?
This touches on a second, highly distinct function of the DLS outcome feedback. The researchers identified a specific, separate subset of neurons in the DLS that do not fire during the run, and they don't fire at the start or end brackets.
When do they fire?
They only fire about half a second after the entire run is completely over and the rat is evaluating the result.
The performance review neurons.
A fitting term. During the early days of learning the maze, about half of these outcome neurons fire enthusiastically when the rat gets a reward, and the other half fire strongly when the rat makes an error, turns the wrong way, and gets nothing.
That makes sense. This is the brain registering success and failure.
Exactly. But as the habit becomes deeply ingrained over weeks of repetitive overtraining, a biological silencing occurs.
A silencing.
Yes. The neurons that signal errors, the ones designed to yell, "We messed up, there's no reward here," drop their firing rate to near zero. They almost completely shut down. Meanwhile, the neurons that signal success and reward consumption remain highly active.
That is deeply unsettling. The autopilot actively stops caring about mistakes.
It does.
It literally mutes the error signals to maintain the structural integrity of the habit loop. The system just assumes the chunk of code works perfectly every time.
And this biological muting of error signals is the exact neurological reason why bad habits, whether it's nail biting, smoking, or doom scrolling on your phone, are so incredibly difficult to break. Your brain's structural architecture in the basal ganglia is actively suppressing the sensory information that would normally tell you the behavior is failing.
Okay, but humans aren't entirely trapped. I mean, we do break bad habits. We learn complex new skills and correct our courses.
We do, yes.
If our deep habit circuits, the DLS, are actively silencing the error signals once a routine is locked in, how do we ever initiate change? We have to have some mechanism that fights back against that subcortical muting.
We do, and it requires shifting our focus away from the basal ganglia and up into the higher cognitive areas of the cortex. To break a habit, you must possess a cognitive mechanism that forcefully processes mistakes.
Okay, so top-down approach.
Exactly. This brings us to the neuroscience of mindset, specifically examining the work of Jason Moser and his colleagues in 2011 on growth mindset versus fixed mindset.
Most of us are familiar with Carol Dweck's famous psychological theories on mindset, right? A fixed mindset is the belief that your intelligence and inherent abilities are static, basically carved in stone.
You believe you're either a math person or you aren't.
Exactly. And a growth mindset is the belief that your abilities can be actively developed through effort, learning, and failure. But what I find so compelling about the Moser paper is that they didn't just give people a psychological questionnaire.
No, they went much deeper.
They put EEG caps on their heads and measured the real-time electrical signals propagating across the cortex when they actually made mistakes.
Because mindset is not merely a positive attitude. It is a framework that alters the physiological way your brain processes errors.
How did they test that?
In their experimental design, Moser's team had participants perform a flanker task, which is a tedious, fast-paced visual sorting task designed specifically to make people slip up and make errors. While they were doing this, the EEG recorded an event-related brain potential called error positivity, or the P wave.
Let's define that. What exactly is a P wave?
The P wave is a specific electrical signal that peaks roughly 200 to 500 milliseconds after you commit an error. It originates primarily from the anterior cingulate cortex, or ACC, a region situated deep in the frontal lobe that acts as an error detection and conflict monitoring center.
So it's an alarm bell.
Exactly. The P wave reflects conscious awareness of the error. It is the physical manifestation of the brain allocating conscious attention to the mistake, essentially broadcasting a signal across the cortex saying, "Pay attention, a massive prediction error just occurred."
And Moser found that individuals who self-reported possessing a growth mindset showed a significantly enhanced P amplitude compared to those with a fixed mindset.
Yes, their error signals were physically larger and electrically stronger.
The amplitude difference was stark in the charts. But what is truly important is the downstream behavioral effect of that enhanced signal, right?
Absolutely. The individuals with the larger P amplitude didn't just register the mistake and feel bad about it. The massive size of that signal directly mediated their ability to rebound.
Rebound how?
Following an error, the growth mindset individuals demonstrated superior accuracy on the very next trial. They allocated more neural resources to staring the error in the face, processing exactly what went wrong, and functionally adjusting their behavior.
I want to linger on the implications of this for a moment. If we think about how we operate in schools or corporate environments, we often try to hide our mistakes or brush past them quickly to protect our ego.
That's very common.
A fixed mindset is basically surrendering to the DLS. It's letting the basal ganglia's habit loop run blindly and allowing the brain to ignore those subcortical error signals, because consciously admitting an error feels like a threat to your static identity.
That's a great way to put it.
But a growth mindset is like a manual override. It is forcing the anterior cingulate cortex to generate a massive electrical wave, flag the mistake, hold it in working memory, and force the prefrontal cortex to rewrite the underlying code.
It is a vital override mechanism, but we have to understand why the fixed mindset exists in the first place.
It's not just a character flaw.
No, a fixed mindset isn't just laziness. It is a highly efficient energy-saving strategy employed by the brain. Acknowledging an error and rewriting a neural pathway is incredibly metabolically expensive.
Back to the energy budget.
Exactly. The brain would rather rely on a slightly flawed habit than burn the energy required to change it. A growth mindset forces the brain to spend that energy.
It's a philosophical shift rooted in biology. Mistakes are not a reflection of a lack of inherent ability. They are a literal biological necessity for neuroplasticity.
Without the error, there is no change.
The brain must generate that strong pay error signal to trigger the release of the neuromodulators like dopamine and acetylcholine that are required to rewire the synapses. If you don't generate the signal, you don't get the neuroplastic adaptation. You stay permanently stuck in the old habit chunk.
Exactly. The electrical signal is the mandatory catalyst for structural change.
This concept of neuroplasticity, The brain physically rewiring its synaptic connections based on learning from errors is incredible. But our sources take this concept of adaptation to a level that is vastly deeper than just electrical signals and synapses.
We are going to the molecular level now.
Right. We are talking about how our daily environments, our habits, and our stress levels literally change the physical structure of how our DNA is read by our cells. We have to dive into epigenetics.
Epigenetics is arguably the most critical frontier in understanding the biology of human resilience. To lay the molecular groundwork, your DNA sequence, the sequence of A's, C's, T's, and G's you inherited from your parents, is essentially fixed. It is the hardware.
The actual code doesn't change.
Correct. But every cell in your body contains the exact same hardware. A neuron in your prefrontal cortex has the same DNA as a skin cell on your arm.
Which is wild to think about.
It is. So the cells require a software program to tell them how to read that hardware. Which specific genes should be turned on to build a neuron? Which genes should be permanently silenced? Epigenetics is that software layer.
The word itself, epigenetics, translates to above genetics. It's the packaging material wrapped around the DNA.
That is correct. The mechanisms involve highly specific chemical modifications to the DNA molecule itself or to the structural proteins that the DNA wraps around. The three primary mechanisms discussed in the literature are DNA methylation, histone modification, and non-coding RNAs.
I understand the textbook padlocks analogy for DNA methylation, but can we break down the actual physical mechanisms of these three? Let's start with methylation.
Certainly. Imagine a long stretch of DNA containing the base instructions to build a specific receptor in the brain. DNA methylation involves enzymes attaching tiny chemical tags, methyl groups, consisting of 1 carbon and 3 hydrogen atoms directly onto the cytosine bases of the DNA molecule.
Okay, so physical tags attached to the DNA.
Yes. When a promoter region of a gene is heavily populated with these methyl groups, they physically obstruct the cellular machinery, specifically RNA polymerase, from binding to the DNA.
So if the methyl group is there, the RNA polymerase just bounces off.
Exactly. It physically obstructs it. If RNA polymerase can't bind, the gene cannot be transcribed into mRNA. Therefore, high methylation generally turns a gene off, silencing it. Conversely, if enzymes remove those methyl tags, the DNA is accessible and the gene is turned on.
Okay, that makes sense. And what about histone modification?
Well, your DNA is roughly 2 meters long if you stretch it out, but it has to fit inside a microscopic cell nucleus.
Right, it has to be packed really tight.
To accomplish this, the DNA is tightly spooled around clusters of proteins called histones. If the DNA is wound too tightly around the histone, it cannot be read. Histone modification involves adding chemical tags, like acetyl groups, directly to the histone tails.
And what does the acetyl group do?
Acetylation alters the electrical charge of the histone, causing it to loosen its grip on the DNA. The chromatin relaxes, exposing the gene so it can be actively transcribed.
And what about the third one, non-coding RNAs?
These are molecules of RNA that are transcribed from DNA but never translated into actual proteins. Instead, they float in the cell and can intercept and bind to messenger RNA, essentially destroying the message before it ever reaches the ribosome to build a protein. They act as fine-tuning volume knobs for gene expression.
The truly paradigm-shifting part of this science is that our lived experiences determine where those chemical tags are placed.
That is the crucial takeaway, yes.
The paper from Demelza Smith, Michael Plous, and others outlines a comprehensive conceptual model for how epigenetics creates long-term resilience. They break it down into phases. Phase 1 is the initial establishment, your baseline genetics, and the potential intergenerational transmission of epigenetic marks from your parents.
Yes.
But then we hit Phase 2, environmental modification. The epigenome is highly plastic, especially during early childhood development.
This phase demonstrates how profoundly our early environment dictates our biological trajectory. For a long time, the bulk of epigenetic research focused almost exclusively on trauma, how severe early life stress places methylation marks that permanently increase vulnerability to psychiatric disorders.
Right, we always hear about the biology of trauma.
But this modern model focuses heavily on protective factors. It looks at the biology of positive environments.
So the biology of care and nurturing is just as physical as the biology of trauma.
The preclinical models, particularly those involving rodents, illustrate this beautifully. Michael Meaney's classic research with rat mothers showed that mothers vary naturally in how much they groom, lick, and nurse their pups.
Some are more attentive than others.
Exactly. High-quality maternal care, which involves intense tactile physical contact, causes a literal, measurable epigenetic shift in the brains of the offspring. It significantly decreases the methylation of the NR3C1 gene in the hippocampus.
Let's trace that mechanism. What does the NR3C1 gene actually do, and how does licking a rat alter it?
The NR3C1 gene provides the genetic blueprint for constructing glucocorticoid receptors. These receptors are the core component of the brain's negative feedback loop for stress.
Okay, the stress loop.
When a rat encounters a threat, its adrenal glands pump out cortisol or corticosterone in rats. When the threat passes, that cortisol travels to the hippocampus, binds to the glucocorticoid receptors, and signals the brain to shut down the alarm system.
So more receptors mean a faster, more efficient shutoff valve for the stress response.
Precisely. The intense tactile stimulation from the mother activates serotonergic pathways in the pup's brain. This serotonin cascade activates transcription factors, specifically one called NGFIA, which bind to the NR3C1 promoter region and actively recruit enzymes to strip away the methyl groups.
Oh, wow.
By removing the methyl padlocks, the maternal care permanently unlocks the gene. The pup produces vastly more glucocorticoid receptors. As a result, their brain is highly efficient at detecting circulating stress hormones and rapidly shutting off the physiological alarm. They mature into calm, highly resilient adults.
The mother's physical touch literally edits the software of the stress regulation gene.
It does.
And we see these same protective epigenetic markers translating to humans. The sources indicate that high-quality parental care in humans, factors like sustained breastfeeding, skin-to-skin contact, and responsive parenting, are strongly associated with decreased methylation of the human NR3C1 gene.
Yes, the mechanism is conserved.
But they also mentioned decreased methylation of BDNF.
Brain-derived neurotrophic factor. This is a critical protein often described as miracle-grow for the brain. Unlocking the BDNF gene promotes neurogenesis, the growth of new neurons, and enhances synaptic plasticity. It makes the brain structurally adaptable.
It's amazing.
Concurrently, we observe that high-quality care increases methylation on genes like TNF, which encodes for tumor necrosis factor, a major pro-inflammatory cytokine.
So the positive environment is stripping the padlocks off the genes for stress recovery and brain growth, while simultaneously placing heavy padlocks on the genes that drive chronic systemic inflammation.
That is the exact molecular signature of resilience. Furthermore, studies on epigenetic clocks, algorithms like the Horvath clock, that measure the accumulation of methylation across the entire genome to determine biological age, show that enriched environments, regular cardiovascular exercise, and nutrient-dense diets literally lower a person's epigenetic age.
Meaning what exactly?
Their cellular machinery ticks slower than their chronological age.
Okay, so a supportive, enriched environment places beneficial epigenetic marks on our DNA. That aligns with our intuitive understanding of health. But the sources introduce a concept called the stealing effect, or stress inoculation.
This is where it gets complex.
Yeah, because this contradicts almost everything we generally assume about the toxicity of stress.
The stealing effect is a fascinating biological paradox. In modern discourse, we frequently categorize all stress as inherently toxic and damaging. However, rigorous epigenetic research suggests that exposure to mild, predictable, and manageable stress can actually confer a profound protective effect.
Like a vaccine.
It acts exactly like an immunological vaccine, but for psychological trauma.
I want to dig into the Brazilian cohort study mentioned in the text, because the findings are wild. Researchers tracked children who experienced prenatal maternal stress, meaning their mothers were exposed to severe stress, specifically violence, while the child was still developing in the womb.
The hypothesis would naturally be that these children would be permanently disadvantaged.
Right.
But the researchers discovered that children who experienced maternal violence prenatally actually exhibited increased psychological and physiological resilience to postnatal maternal violence compared to children who only experienced violence after they were born.
That is so counterintuitive.
It is. The exposure to the stress hormones in the womb physically altered the DNA methylation patterns on key stress response genes, most notably FKBP5 and the NR3C1 gene we discussed earlier.
Wait, let me make sure I'm synthesizing this correctly. We normally conceptualize stress as a corrosive acid that breaks the biological system down. But you're telling me that a moderate dose of stress early on acts like a biological vaccine. The fetus senses the high cortisol levels from the mother, realizes it is being born into a hostile environment, and epigenetically alters its DNA expression to build biological armor on its cells before it even takes its first breath.
That is an incredibly accurate interpretation. The FKBP5 gene encodes a co-chaperone protein that regulates how sensitive the glucocorticoid receptor is to cortisol. By altering its methylation, the developing brain is fine-tuning the sensitivity of its alarm system to match the harsh environment it anticipates entering. If we connect this to the broader philosophical picture, it forces a complete redefinition of resilience. Resilience is not simply the absence of trauma. It is not an empty, pristine state of nothing bad ever happened to me.
Right.
Resilience is a highly active, metabolically expensive biological process of adaptation. The organism senses a stressor, processes the error signal, and epigenetically tweaks the hardware to survive that specific challenge in the future.
It's the organism saying, the world is difficult, let's unlock the exact genetic tools we need to navigate it.
Exactly.
And the most hopeful revelation in this epigenetic literature is that these chemical tags are not permanent. They are reversible. The sources highlight studies showing that effective, evidence-based psychotherapy for conditions like PTSD can actually reverse the aberrant DNA methylation marks on genes like SLC6A4 and FKBP5.
It proves that psychotherapy is not merely talking it out or venting emotions. It is a targeted biochemical intervention.
How does talking change DNA?
Through the prolonged cognitive effort of confronting traumatic memories in a safe environment, the patient generates new neural firing patterns. These new firing patterns trigger intracellular signaling cascades that physically remove the biochemical padlocks from the DNA inside the nucleus of the neurons. The biological scarring of trauma is not a permanent life sentence.
That is incredibly profound. But there is a glaring catch to this entire framework, and we must address it to be intellectually honest. We've said that stress can act as a vaccine, the stealing effect.
Yeah.
But obviously, clinical data shows that isn't true for everyone. Two individuals can experience the exact same severe traumatic event. One person experiences the stealing effect, adapts epigenetically, and bounces back stronger. The other person experiences the exact same physiological stressor and develops debilitating chronic PTSD.
That is the core puzzle of psychiatry.
If the mechanism is the same, why is the outcome so drastically different?
The divergence in those outcomes lies in the complex interaction between the dynamic epigenome we just discussed and the underlying static genetic hardware that the individual was born with. This brings us to the final piece of the puzzle: the genetics of environmental sensitivity.
Why some people are genetically wired to react differently to the world.
Exactly. For decades, the fields of psychology and psychiatry relied heavily on a framework known as the diathesis stress model. You can conceptualize it as the broken vase theory of mental health.
The broken vase theory.
Yes. It posited that certain individuals inherit fragile or defective genetic variants. If you take a person with these fragile genes and place them in a highly stressful, adverse environment, they will break and develop severe psychiatric illnesses.
Okay, that makes sense.
However, if you place them in a highly supportive, low-stress environment, they will function normally, just like a person without those genes. They simply carry a hidden vulnerability.
Right. So under that traditional diathesis stress model, possessing these sensitive genetic variants was viewed purely as a biological liability. You're essentially carrying around a ticking time bomb, just waiting for a bad day to trigger a depressive episode.
That was the consensus. But recent large-scale genetic and behavioral studies have completely upended that dogma by introducing the concept of vantage sensitivity.
Vantage sensitivity.
Yes. Researchers began to realize that these so-called fragile risk alleles do not exclusively make an individual vulnerable to adverse environments. They actually make the individual exceptionally, disproportionately responsive to positive environments as well.
Break that down with a specific genetic example from the research.
Let's examine the SLC6A4 gene, which is responsible for encoding the serotonin transporter protein. This protein acts like a vacuum cleaner, sweeping up excess serotonin from the synaptic cleft after a neuron is fired.
Okay.
There is a specific variant of this gene, the short allele, that results in fewer transporters, meaning serotonin hangs around longer in the synapse. Historically, this short allele was famously labeled the depression gene.
Because it was linked to hyperreactivity in the amygdala, right?
Exactly. But under the new vantage sensitivity framework, researchers studied youth carrying this highly sensitive short allele to see how they reacted to the quality of their parenting.
What did they find?
Under the old diathesis stress model, you would predict these children would show massive spikes in behavioral and emotional problems if exposed to harsh parenting. But the longitudinal data was nuanced. While they did struggle in negative environments, the most significant finding was their reaction to good environments. The youth with the sensitive variant showed a massive, disproportionately positive developmental response to highly supportive parenting, far exceeding the baseline resilience of children who carried the normal, supposedly robust genetic variant.
Let me make sure I fully grasp the magnitude of this. What society and old-school psychiatry used to pathologize as being fragile or overly sensitive is actually a profound biological superpower.
That is what the data suggests.
You aren't just a delicate flower waiting to be crushed. You are biologically constructed as a high-gain antenna. Your system is designed to pull in significantly more developmental benefit from positive inputs like a loving home, effective psychotherapy, or a daily meditation practice than a person with resilient genetics.
That is the exact paradigm shift that Vantage sensitivity introduces. If you possess these highly sensitive, highly plastic genetic variants, your entire biological system is inherently designed to intimately soak up the nuances of your environment. Yes, an intensely toxic environment will harm you deeply because you are absorbing it. But a nurturing environment, an enriched cognitive setting, will propel you to levels of flourishing, empathy, and psychological resilience that a genetically less sensitive person might be biologically incapable of reaching.
It flips the entire narrative of vulnerability on its head.
Yeah.
If you have been told your entire life that you are too sensitive or that you let things affect you too deeply, modern molecular biology is essentially saying, yes, your microphone is turned all the way up.
Yes.
But that simply means you possess the neurological capacity to hear the beautiful music better than anyone else in the room.
It is a highly empowering reframing of our biology. It demonstrates that true resilience is not a single trait. It is a highly complex, polygenic, and dynamic state of being.
A constant interaction.
Exactly. It is a continuous, lifelong dance between the inherent genetic code you inherited, the epigenetic modifications layered on top of that code by your daily experiences, the automated habits your dorsolateral striatum chunks together, and the cognitive mindset practices you actively employ to process your failures.
So what does this massive scientific journey mean for you?
Yeah.
We have traversed the hidden microscopic architecture of the mind today.
We covered a lot of ground.
We really did. When you sit quietly on your couch, attempting to do absolutely nothing, your brain's default mode network is burning massive amounts of metabolic energy, actively weaving the narrative story of who you are, constantly pulling from your past traumas and projecting into your future anxieties.
Right.
When you stand up and physically interact with the world, your basal ganglia and infralimbic cortex are busy writing code, chunking your behaviors into unbreakable automated blocks, muting your error signals so you don't have to overthink your reality. When you fail or make a critical mistake, your anterior cingulate cortex generates error positivity signals, giving your higher prefrontal cortex the necessary catalyst to wake up, allocate attention, and physically rewire that code.
That's the manual override.
Exactly. And through it all, through every single hour of every single day, every hug you receive, every rigorous walk you take, and every challenging therapy session you endure is physically altering your epigenome. You are actively moving microscopic chemical padlocks around your DNA, writing and rewriting resilience into your very biology.
It is a stunning, deeply interconnected system, and I always emphasize to people, biological knowledge is most valuable when it is consciously applied.
Applied how?
We have a tendency to view the adult brain as a static organ, a finished architectural product that slowly degrades over time. But every single mechanism we have explored today, from the functional plasticity of the DMN under psychedelics or meditation, to the electrical P waves of a growth mindset, to the epigenetic unlocking of the NR3C1 gene, proves unequivocally that the brain is a lifelong, active construction site. You possess profound biological agency over how that construction proceeds.
And realizing that biological agency is what I want to leave you with today. If your resting brain uses the incredibly powerful default mode network to build your core identity, and your external environment uses epigenetic mechanisms to physically turn your DNA on and off, are your default thoughts truly your own? Or are they merely the biological echoes of the automated habits, the stressful environments, and the internal narratives you have unconsciously chosen to surround yourself with? Think very carefully about what you are feeding your autopilot today, because the exact moment you sit down, put your phone away, and try to do nothing, that is exactly when your brain goes to work.